We're not doing science for ourselves. We're doing this for you. When you have a question and you want to answer it, the perfect person to do that is a scientist.
This is our humble hemp patch.
5,000 years of medical cannabis use.
We're learning about other cannabinoids.
Marijuana is grown in every state in the Union.
I'm Lex Pelger, Director of Education at CV Sciences, and this is the Lex Files. No two cannabis plants are exactly the same, even when they have the same genotype. The research of Dr. Anna Schwabe is all about finding out why genetically identical plants in the same laboratory settings will produce a different level of THC, CBD, and other compounds. She's a prolific cellular and molecular biologist with a long, diverse resume. She joins us today to discuss genotypes, phenotypes, epigenetics, how to identify cannabis cultivars by their smell, and why it takes so much training to draw proper pictures of pot plants. Please welcome today's guest, Dr. Anna Schwabe. Hello, everybody. Welcome to today's show, and I'm very pleased to be here with Dr. Anna Schwabe. Thanks for joining us.
Hi, it's nice to be here.
Before we get into all of your work as a scientist, I was curious what got you onto this path and all the different fields of science that you're familiar with, and what did you want to be when you were little?
So I distinctly remember telling my mom when I was little that when I grew up, I wanted to be a biker chick with half pink hair and half green hair. And she says that I got half of that right.
I'm looking at a head full of pink hair right now.
But I don't know what I wanted to be when I was little. I've always enjoyed, inquiring, answering questions, doing puzzles, things like that. So I guess that's why science is cool. I started out in anatomy and physiology thinking that I might want to be a vet someday. So I did 5 years at CSU with anatomy and physiology, thinking that I was going to go to vet school, but my grades weren't good enough. And it turns out I really like animals. And to be a vet, you got to do some pretty big soul searching because, putting down animals and stuff like that, I don't think I'd be able to do that very well. So, so then I switched gears into cellular and molecular biology, and that's kind of where I've been. So,
And so how did that path start for you? Because, on your site, you list, A&P, molecular biology, stats, evolution, speciation, genetics, and science communication illustration. So what did your early schooling look like to get you in all these different fields?
So, like I said, I started with anatomy and physiology of animals and have 5 years of background in that. And then I took a hiatus from school after 5 years. I didn't actually graduate from CSU with my bachelor's. I kind of messed around a lot and that sort of thing. But then when I came back to school, I started in cellular and molecular biology, and I took like another anatomy class. I have so much anatomy behind me. And then when I came to graduate school, as part of graduate school, we have to teach. And so I was like, I know a lot about anatomy and physiology, and a lot of people don't want to teach that because it's a lot. So I ended up teaching anatomy and physiology, but I was doing plant studies, and people are like, why is somebody who does plants teaching anatomy and physiology of humans? So it turns out a lot of plant people teach anatomy and physiology. I don't know why that is, but so that's why I have so much anatomy and physiology. That's my teaching background. And then as part of the doctoral degree that we have at UNC, all doctoral degrees at UNC are education degrees. So I actually have a degree in biological education, and then the research portion of that I wrote my dissertation on was actually, a whole nother thing. So science students in particular, biology students, we all have our own research, but the PhD actual degrees in biological education. So we have to learn how to teach how to learn, do course development. I taught conservation biology as part of my degree. So yeah, full-on teaching and research and doing all kinds of stuff.
Wow. Is it difficult to wear those two different hats? Because in some ways science, it seems silly because these people who are great researchers and spend all their time in the labs often aren't the best people to be teaching.
Yeah, and I totally get that. And that is, so I know a lot of PhD students at UNC feel like it's a lot of work to be doing research and teaching and then doing the education degree as well, because we have to take a lot of classes for that. I didn't find it particularly hard. I mean, I'm pretty good at time management, and I didn't find it particularly hard. And I think I'm a pretty good teacher.
I suspect, from what I've seen online, and you'll see some links in the episode notes.
Yeah. But I mean, some people are not very good or they just don't care. You know, it's part of something that we have to do to get through the degree, and so they just, you know, it's just something that you have to do, and, you know, just get through it. I don't want to be a teacher. I've never wanted to be a teacher, but I do know how to teach. So I guess if something came up, I would have that to fall back on. But, you know, I mean, every day I'm teaching somebody something, you know, just through talking about my research and all that. So.
And speaking of your research, can you tell us about what your dissertation was on and what it took to actually get that approved when cannabis is still so verboten in so many ways?
Yeah. So it was funny because when after I worked at the Denver Botanic Gardens and my position finished, I had a really hard time finding a job. And I went to an interview at, it was one of the chemical labs down in Denver, and I interviewed with them, and they thought I was great, but they ended up hiring somebody over me that had a PhD. And I was like, well, I could get a PhD. So I had some really interesting ideas, and I approached my advisor that I had for my undergraduate, or for my master's work, and brought this idea to him, you know, cannabis, we could do this. And he said no. He said, I don't want to be that guy. And every time I saw him, I was like, come on, you know you want to do it. And it was actually his wife that talked him into it. She said, somebody has to be that guy. Why don't you be that guy? And it's nice because UNC is a small enough school that, you know, the dean and you know, everybody was behind me saying, yeah, we can totally do that. You just can't have it on campus. And I was like, well, I don't have to have it on campus. I can do partial extractions off campus and then, you know, do all the analysis and PCR and stuff on campus. And they said, as long as you're just working with DNA and you don't bring any, you know, material that could be smoked, you can do this. And so, MIT finally said yes. And it started out as a project that was going to be looking at hops. And then as a side note, I was going to do cannabis since they're related. That flipped 180 degrees to all cannabis, no hops, which was kind of funny. And the whole premise for my dissertation changed and evolved. And even though I'm graduated, I'm still working on one of the projects that I had developed. But yeah, so the first idea was, you know, that first paper that I put out with the variation within strains. And then from there, it just kind of took off, and I did all kinds of other stuff.
And one of the talks that you gave on sources of variation in cannabis, you called joint forces.
I did.
Which I thought was good. That was really good. A lot of these weed jokes I don't like, but that one's too perfect. So can you talk about genotypes versus chemotypes and how this all gets affected by the epigenetics?
Yeah, so there's the big equation that we have when we look at organisms, right? So when you look at an organism, whatever they look like on the outside, and that includes, you know, terpene and cannabinoid production, we can measure those. That's all part of the phenotype. So that's things that you can look at. You can say, that's what that is, that's green, that has flowers, that smells like this. So that's all phenotype. So the physical observational characteristics of an organism. And the phenotype is a product of the genotype, which is the DNA, plus the environment. So they've done twin studies where two twins that have identical genotypes end up being quite different in adulthood due to, you know, different lifestyles, things like that. And so when you take that genotype portion out of the equation, like if you have exact genotypes, you can kind of make the assumption that any differences in their physical appearance or the physical characteristics is a product of the environment, because you know you started out with identical genotypes, right? So with cannabis and the joint forces, I looked at, taking two identical plants, getting, genotyping them to make sure they're identical, and then looking at their phenotype. So their chemotype is a perfect thing to look at, because if you don't have the plant to look at, if you just go to the dispensary and you buy a sample, you've got the bud, which has a chemotype. You can look at the chemotype. If they're all genetically identical, they should all have the same chemotype, but we know that's not true. So that means there's some environmental things feeding into this, and if they're from different dispensaries, presumably these identical plants have been grown under different conditions, because every master grower has their little tips and things, and watering regimes and nutrients and all that. So of course they're going to be a little bit different. However, if you've got identical plants, clones, and you're growing all of them under the same conditions in your grow facility or your incubators or whatever, and there's no environmental differences, and they still turn out different, how does that happen? And so, I'm looking into the epigenome, which is not the actual DNA. That stays the same, but there's marks on top of that DNA that can be added or removed, allowing the plant or the organism to express genetic traits differently. So that can lead to a new phenotype, even though you've got the same genotype and the same environmental conditions. And I think some of the epigenetic changes that occur could be due to stress. Let's say stress from cutting, from cloning, from regrowing. But, nobody's really done the work yet. So it's an interesting thing to think about.
Because one of the things I always think is fascinating about epigenetics is that we used to call that junk DNA, and it turns out to be the control DNA for the genes that are actually turned on. And in humans, if you hurt somebody or someone hurts themselves, something like smoking, genes get permanently turned off. So you might still have those genes, but since they're not working, you might as well not have them. And so it's interesting that this same thing is happening with plants, and it seems like it's more unknown and harder to measure.
Well, so with plants, we people can, if we're in a stressful environmental condition, like if it's too hot, we can just get up and leave. A plant can't do that. So if there's something in the environment that is stressing a plant out, they have to do something in order to adapt very quickly. And the general adaptation, through mutation events and, all the things that lead to speciation, and that happens really slowly over time. And if you're a plant that has to adapt quickly, then you've got to have something else in your toolbox to make that happen. And so plants are actually pretty good at using epigenetic pathways to kind of, and it's not like they're thinking, oh, I should upregulate this, heat gene. Like they just throw it out there. Whoever survives through that stressful condition, they can pass on to their next generation those epigenetic marks, so that their offspring can also survive through what they would assume to be an ongoing difficult condition. So that's the cool thing about plants and epigenetics, is it's something that's heritable, but it's not as slow of a process as adaptation is.
And speaking of evolution, what's your opinion on the evolution of the cannabinoids themselves? It seems like there's a lot of different theories out there. Do you have a favorite on why the plant started making these expensive fatty molecules?
That's a good question. I've heard some of the, people talk and trying to figure out what cannabinoids are, why they're present, that almost all organisms except insects have an endocannabinoid system. So why is it that insects don't have it and humans do? There's that coevolution theory where the plant evolved along with humans, and we kind of have this, they help us and we help them get around the world to different places, because plants, they can't move themselves. So it might be some sort of, symbiosis situation where we help them, they help us, but I don't really know. And then as far as, what the plant is doing, it's mostly been controlled by us. And the way that we've been selecting our favorites for thousands of years. So, one of our favorite things about cannabis is THC. Maybe there's so much THC in there because that's what we've chosen, not necessarily that it's beneficial for the plant to have THC. Same thing with terpenes. We smell it. Ooh, I like the way that one smells. I'll keep that one. Ooh, that smells like stinky feet. Get rid of that one. So I think also the terpenes, although, some people say that it's to keep away insects or for, some sort of defense, I think that we've been messing with it so long. Really, the whole thing is just us. We've selected for all of these things.
And speaking of, that's some of your really cool research to read about, is trying to figure out what we've done to the genes of this plant, especially in the recreational market of the United States, which is kind of a weird hybrid of different regions of cannabis. And so with your work, how would you fill in this gap in genetic testing and the many wrong names for marijuana strains out there?
There's a lot to unpack there. So, what I look at, so there's genetics, as a whole field, and a lot of people are looking for the genes that control for this or help the plant express this because they want to have bigger flowers or, higher THC content. I don't actually look at genes. I look at the junk DNA, the stuff that's in between the genes that doesn't actually help the plant code. It doesn't code for anything. It's neutral genetic markers, and that way you can get sort of a better idea of the history of the plant, who's related to who, without bringing in these things that have been purposely selected for, and that's kind of like an artificial signal coming through. So I can get a better look at what the plant's actually doing. And so using those neutral genetic markers, we can look and we can test for, are they identical? Because the answer is either yes or it's no. If they're not identical, it's not always possible to say what it is, to identify it as, oh, that's not Blue Dream, that's actually sour diesel. What I can say is that's not Blue Dream. That's not the same as all these others. It's different. How is it different? I don't know. It just is. It's not what these other things are. And so that's where you can look and you can say, okay, with the names, we do have some misnaming or some misidentification, or perhaps in dispensaries they're putting another name on something that isn't selling to make it move quicker off the shelves. So I think we could use genetics to kind of look at that a little bit more and give some more consistency to the industry. And then I like the strain names. I mean, if I was a breeder, I would be kind of bummed if they went to, a naming system that was like, this is G5496, and it has, terpene content, A2. I'd be like, what? I don't want to talk about my babies like that. I think the naming is kind of cool. And people inherently need to categorize things, and we need to have a way to talk about things. If I say Blue Dream to you, you know exactly what I'm talking about. You know that there might be variation among dispensaries, and that, Blue Dream out in California might not be the same as Colorado, but you know what I'm talking about, at least. If I came up to you and I said, man, I found this really awesome strain in G5942 with the terpene profile A2, and you'd be like, I have no idea how to process that information. You know what I mean? So I think the strain names are important just for communication purposes even, and even the whole sativa indica thing where scientists are like, no, we haven't found it, there's no support for it. But people, customers, consumers are like, yeah, there is a difference, and we can tell the difference. And what that difference is might be different to you than it is to me, but you might know, oh, I hate sativas, they make me feel weird. And I might be like, oh, I love sativas, they're my favorite. We still know what that sativa means to us. You know what I mean? So whether or not science has found it yet doesn't mean it's not there. They just haven't figured it out yet. So, I think this is all really important stuff for people to be able to communicate with each other. So that's what I think about that. That was the short answer.
It's a good one. For years, I've been writing about cannabis and the endocannabinoid system. I gathered people's stories from around the world. I learned about the long human history with hemp, and I interviewed scientists about the importance of our own cannabinoids. But at the time, the positive effects of CBD were only starting to dawn on me. It took me seeing the impact that CBD-rich hemp extracts made on the health of my cousin, and the comfort it gave to some of my elderly relatives, especially the ones who are always against pot. Since then, the many stories I've heard from people using CBD made me a believer in its potential. That makes me proud to now be working at CV Sciences, because I know firsthand how hard they work to produce the finest quality CBD-rich hemp supplements. I'm proud because I know we lead the industry in research and education, proud because I know we make excellent CBD oil from true agricultural hemp, and I'm most proud because I know our products make a difference in people's lives. At pluscbdoil.com, use a coupon code Lex Files for 20% off to see for yourself. And actually, the next question then continues with terminology with cultivar versus strain. I mean, as a plant geneticist, how do you stand on this? That strain isn't a typical botanical word.
Well, cannabis isn't a typical botanical plant. Like when we develop a new tomato, we do lots of back crossing and inbreeding to breed out all of those traits that we don't want in our brand new tomato strain or cultivar, and then when we go to register that new tomato and we're going to call it, Anna's Delight or whatever, that is a stable seed line. You'll get the same individuals anytime you pick up a packet of Anna's Delight from Lowe's. You can expect to get those kind of tomatoes. With cannabis, we don't really do a whole lot of sexual breed. Like, we don't go to seed, we don't grow from seed that often, and the seeds that are available are not that stable. And there are growers and there are breeders who take the time and the work to back cross and inbreed for 12 to 14 generations to get that stable line. But most of the strains, and I'm going to call them strains, out there are a one-off cross of two things that were hybrids themselves. So we've got hybrids of hybrids of hybrids. You know what I mean? And so I feel like the term cultivar, although it's more botanically correct, can't necessarily be applied to cannabis types because they don't come from the same sort of place. Does that make sense? It's just sort of crossing things that have been crossed, and a lot of them are fleeting. You grow up 100 seeds, you pick the ones you like, that's the mother plant. Once that mother plant is gone, you're either doing clones of clones, which can be a little tricky, or once that mother dies, that strain's gone. It's no longer available, it's done. So that doesn't happen with cultivars, right? So that's how I feel about that. So I'm going to keep using the word strain because I like it.
That's good. And speaking of looking at what's actually out there for real people, you did work looking at the National Institute for Drug Abuse and the cannabis that they produce at the University of Mississippi and compared it to the commercially available cannabis out there. What did you find?
The product coming from the National Institute on Drug Abuse is not genetically close to what is available to general consumers in the dispensary. It's actually closer genetically to wild growing hemp and cultivated hemp. Yeah, so I went and I got collected hemp samples from herbaria. So things from 1976, things from 1969, things from, last year, ditchweed growing in, wherever. And then I also had some very nice hemp farmers who sent me samples and said, here, this is, industrial hemp or, high, it's hemp. And then, so it was more closely related to the closest relative that in my sampling set was a wild collected hemp, we assume it's hemp, from 1969.
Wow. So they're basically growing hemp ditchweed
Well, so
for patients.
I mean, there's lots of ways this signal could have, I don't know how to say this, but like the hemp plants that I have a very strong single genetic signal. And the samples from NIDA have that same strong genetic signal. Now they're growing outdoors in Mississippi. And somebody pointed out to me that they also grow hemp in Mississippi. And hemp is a prolific pollinator. And it could be that the University of Mississippi's plants have been, over the years, heavily pollinated by outdoor grows. And now what they have is hemp.
Wow.
And without genetic tests, you wouldn't really think about that, and you wouldn't really know about that. The other thing is that, maybe back in the 60s and 70s, all of these plants were much closely genetically related before everything went underground for a long time and all that kind of stuff. It could be it's just really super inbred, and it doesn't have a signal that's the same as anything else. So there's a lot of different reasons why we could be seeing this, but without talking to them and getting answers from them, it's all just guesses. So, yeah, I don't know. But it's definitely not anything like what you or I would be able to put our hands on and use for medicine. So I feel like a lot of the studies are just inherently flawed just because they're not what people are saying. If somebody says, yeah, I use this and it's awesome, it gets rid of my headache. And then NIDA says, well, we've tested it and it doesn't. Well, it's because you're using something totally different, So.
Is it difficult to sometimes see the lack of precision out there from what's being called various strains of cannabis and what's being given by the federal government? I mean, with it being such a complicated plant, what's it like for you trying to tease out all of these different variables that are in each individual?
Okay, so, it doesn't affect me personally. All of my projects, I don't do hypotheses. I don't say, well, I think this is what's happening and then test my hypothesis. I feel like that kind of science kind of makes you biased from the gate. So I literally went into this not having any idea of where. I figured hemp would have its own signal because other people have looked at it too, and hemp is pretty distinguishable from any of the drug types. I didn't know where my high CBD strains would fall. I assumed somewhere between the drug types and the hemp types, which they did, so that was nice. And then the NIDA stuff, we just had no idea what would happen with that. We were just like, well, let's just see who wants to hang out with genetically, and so that's what we found. It's not, and we're using, the markers that we use are not, as I said, they're not expressing genes. They're the neutral markers, and so it gives us a good idea of evolutionary history and things like that. So it was just interesting to see. I wouldn't say it's like frustrating, but it also gives validity to all of these claims from people who have, said, well, things are different when I get it from different dispensaries. Why is that? Well, I answered that question. They are different. Why? We don't know. But now people can see all of these different places where variation could have happened. It could be the seed company. It could have been the clone. It could have been that George didn't actually know that he didn't have Blue Dream, that he had something else. And so now we can start to move towards checking some of these things. Do I have Blue Dream? I can say, yes, you have Blue Dream, or no, you don't. I can't tell you what you do have, but, if you think you have Blue Dream, you bring it to me, I test it, and I say, that's definitely not Blue Dream, and they'll say, what is it? I'll be like, I don't know. What do you want to call it? You know what I mean? So what I want to do with all this stuff is just open people's eyes that it's not as well regulated as other pharma industry. It should be dialed in a little more than it is. And I'm not saying that we need to, put all these restrictions and standards and things like that, but people should know what's actually going on, and that there's a possibility that you'll get something different until we figure it out.
And that's actually a favorite question I like to ask. If we could put you in charge of some of the laws regulating dispensaries, what would you put in place as some of the most important for accuracy and consumer knowledge?
So I've thought a lot about this question, and it's not. I don't think it's fair to ask everybody to have their plants genotyped. I mean, if you want to come up with a brand new strain and call it, the cloud or something, go ahead and do that. That's fine. But what I can see happening is something like the vitamin industry. You go walk into the store, there's hundreds of different kinds of vitamins from all these different manufacturers. Some of them have a certified stamp on them that say, this has been tested rigorously through this company. When you buy this bottle of vitamin C, you'll know what you're getting. Whereas if you buy like the off-brand, hasn't been certified, doesn't really have any testing information, you could buy that if you wanted to, and there's no reason why you shouldn't, but it's not certified. So you could, if you're a medical patient, let's say, if you've got a certified, if you can go somewhere and somebody's certified, yes, this is Blue Dream, then maybe that's for you. If you're just, a recreational consumer out to, test all the different things, then it doesn't matter that it's certified or not. Like, you're just going out and seeing what you like. So I can kind of see it going that way. Yeah.
That's great. 2 different levels, kind of a bottom shelf and a top shelf.
Yeah. And depending on what you're looking for, I mean, if you don't mind a little surprise here and there, then you don't need to buy the certified stuff.
And speaking of finding stuff that you like, you've also presented on olfaction, on smelling in cannabis strains. Can you talk more about that work?
Yeah, so after I did the first variation within strain study, I was contacted by Avery Gilbert, who is a scent scientist. This is his jam, and he's just recently put out a couple of papers on cannabis. But as soon as he contacted me, I was like, I totally know where you're going with this, because I can tell if things are identical genetically, but does that mean that the phenotype is identical? And a perfect way to test that is smell. Testing terpenes can get kind of pricey, but, give somebody a $5 gift card to Starbucks to smell some weed and they'll do it. So that's what we did. So I genotyped some of the most common strains, found genetic identical samples, and then it was a double-blind study, so I knew what the samples were, but Avery didn't, and neither did the participants. And they were all in little jars with a number on it, and basically he would give them a jar, they would smell it, and they would do a check all that apply survey on like an iPad. And so there were 40 different odor descriptors, everything from coffee to skunk to blue cheese, diesel, chemical, flour, all kinds of herbal smells. And so if they smelled 20 of them, then they'd check 20 of them. If all they smelled was skunk, then that's what they checked. And what we found was, so in each lot I had a minimum of 3 samples that were genetically identical and 1 that was genetically very different but had the same name. And in Mob Boss and Blue Dream, the one that was genetically different definitely smelled different than the other ones. Durban Poison was all over the place, even though all 3 of them were genetically identical. And OG Kush, the outlier, the genetic outlier, definitely smelled different and had a distinct cheese aroma, which people picked up on more than is normal. So that was kind of crazy. But yeah, it was really interesting. So yes, people could pick up the genetic outlier probably because it wasn't what it was labeled as. It was something different. But as far as being like, you can smell, you can identify what sour diesel is by its smell. It's the variation in the scents were like all over the place, and I don't know that many people with that special sense. I'm sure maybe there are some people out there, but yeah, as far as identifying what kind of strain it is by the smell, I'm not sure people can do that, but they can definitely tell the difference between things. So that was a fun study.
The nose knows. And the last thing I actually wanted to ask about was your work with illustrations.
Yeah.
And how you got into that.
So I So I've been at UNC. I have 3 degrees from here now. My bachelor's, my master's, and now my PhD. When I was doing my bachelor's, I had to take this is going to be a long story, but it's a good one. I had to take an upper level biology class in order to graduate on time. And I really wanted to take animal behavior, but it was full. And they wouldn't override anybody into it. And the only other class that was available was plant systematics. And I did not want to take that class at all. Like, plants are stupid. Why do I have to do this? But if I wanted to graduate on time, I had to take this class. And it was the first class that this brand new professor that was, totally green to UNC was teaching. And it was Mitt. It was my advisor. But he made it really exciting. And, he gave us a list of 100 must-know plants. And then we had an assignment. We had to go out and collect 20 things that were not on the must-know list, which irked me because I was like, there's like no more plants. Where are we going to find 20 more plants that aren't on this stupid list? Like, I just seriously had no idea about plants. And as part of that assignment, when we turned in our 20 plants, and I think I had like duplicates in there because I really couldn't find that many plants. We had to turn in, we had to identify it, so we had to key it out in one of those field books. We had to do a herbarium label, so we had to write where it came from, who collected it, the date, any pertinent information, and then we also had to do a sketch of the plant. And so when I turned my assignment in, Mitt said, "You're really good at drawing. You should go and check out the botanical illustration classes that they do at the Botanic Gardens." And I was like, "Yeah, that sounds like fun. I could take a class or two, whatever." It's a little bit more intense than that. It turns out it's like a whole other degree. You have to take 13 core classes and then 100 hours of electives to get to get certified in botanical illustration through the Denver Botanic Garden School of Botanical Illustration. And so in between my bachelor's and my master's, I just did that. So that's how I got started on that. And I have a website that has some of the stuff that I've done, and I've done commissions before, but I don't really do it very often. It's usually if somebody commissions me or will pay me to do it, then I'll draw something, but I don't, it's not my favorite past. I don't sit out in the garden and draw, which is kind of funny, but yeah, that's how I got started with that.
That's great. And because it's very classic and elegant work, but you're also putting a new take on it. I mean, I like scientific illustrations, and yours really stand out.
Well, thank you.
So yeah, we'll have a link to it. And actually, the very last question I feel like I should ask, I said it. Could I ask about your tattoo and the molecules on it?
Yeah, well, so these are my study species. I have one on each shoulder, right? So this is my sclerocactus, which was my master's. And on this side, this was drawn by a friend of mine. He didn't draw it for me. He just drew it and used it as his logo. And I was like, I want that tattooed on me. And he's like, yeah, okay, here's the PDF file. You can take it to your tattoo artist. So what it is it's a THC molecule. And then upside down is the CBD molecule. And in between is kind of this twisted ladder that kind of looks like DNA. So it's kind of like a yin and a yang with the DNA and then the THC and the CBD. So it's kind of, I don't know, it's cool. And if you don't know anything about THC or CBD, you would never know what the molecule is. It could be dopamine and serotonin for all anybody knows. But yeah, it's THC and CBD. I didn't want to get a pot leaf because that's so tacky.
The molecules are classic.
Tiny molecules.
Well, Doctor, thank you so much for your time and for your research. It has been really enjoyable.
Yeah, I've really enjoyed doing it, and I really hope that I can keep answering people's questions because, as I said to you before, if we're not doing science for ourselves, it's not because of all the money we get, because we don't get any money. We're doing this for you. When you have a question and you want to answer it, the perfect person to do that is a scientist, and I feel like I'm doing the science that I do for the people. So I hope to keep doing it, keeping it real, and bringing it back to you. So,
well, thank you for sharing, and thank you for your work.
Yeah, thank you.
Thanks for tuning in. To listen to other episodes, find us at pluscbdoil.com/Lex Files. If you have any questions, compliments, or suggestions, feel free to write me at research@cvsciences.com. If you enjoyed the program, please rate us on iTunes and share a link to your social media. It means a lot to us. The Lex Files is produced by Matt Payne. Our chief advisor is Amabel Dela Cruz. The music is by Jake Bradford Sharp. Our sponsor is CV Sciences, maker of America's favorite CBD oil. And remember the coupon code Lex Files. I'm Lex Pelger, signing off.